In Chapter 6, we are going to be taking a look at the cell. And specifically, we're going to be looking at the structures and the functions of those structures inside of a eukaryotic cell. Where we're going to start, though, is by looking at microscopes. And the reason that we start here is because cells are so small, we need microscopes to view them. In lab, you guys are going to be using a light microscope where light is used to pass through a specimen, and then that light magnifies whatever image you're looking at. Light microscopes have three properties: magnification, making an image appear larger; resolution, making an image appear clear; and contrast, making different parts of the image different so we can see them and tell one from the other. Now, if we look at this chart, you can see what the human eye can see without any assistance. This section right here, we're talking about 10 meters to somewhere between 1 millimeter and 100 micrometers. This is the region that we see using a light microscope, so we'll be able to look at animal cells, plant cells, and you'll be able to hopefully see the nucleus of a cell. With our microscopes we're going to be using, we're not going to be able to see bacteria or mitochondria. They are too small. And if we use electron microscopes, which we don't have here, those can go the whole way down to almost the size of an atom. We can see very, very small molecules with these. So we're going to take a look at what electron microscopes are and how they differ from light microscopes here in a second. So light microscopes, we can magnify about 1,000 times an image the size that it actually is. We will be using stain to help enhance the contrast. So we'll be staining your cells so that you can see the nucleus of your cells. But we will not be able to see the other organelles like mitochondrion, ribosomes, Golgi apparatus, because they are too small to be seen by a light microscope. This is a typical light microscope. Again, we're going to be doing a lot more work with these in lab, so I'm not going to spend time going through the different parts of them right now. We will become pros with that shortly. Now on the other side, electron microscopes, they can magnify much, much greater than a light microscope. There are two different types of electron microscopes. Scanning electron microscopes, and they do just what they sound like they do. They use electron beams to scan the surface of a specimen. So when we get these images back, you're looking at a three-dimensional image of the surface of a specimen. If you look at this picture, you're looking at the surface of cilia. Transmission electron microscopes, those specimens are usually thinly sliced and electrons are sent through the specimen. It's very similar to how a light microscope works, but instead of using light, these electron microscopes use electrons. And because we're sending electrons through the specimen, transmission electron microscopes produce a flat image where you're looking at the inside. So it's like a cross-section of different structures. So this picture is the same thing that came from here, but now we're just looking at the inside of it. We would see something like this similar if we use a light microscope, but of course we would have to look at something larger than a cilia. Once we get done talking about microscopes in lab, this will all make more sense, but again, I just wanted to give you a brief overview of the light microscope versus the electron microscope. Probably what's more important is this idea of eukaryotic and prokaryotic. Now, if we could take every living thing on the face of the earth and classify them based on the type of cell that they have, we would end up with 2 or 3 major categories. If we say 2 categories, cells can either be prokaryotic or eukaryotic. And our eukaryotic cells are the ones that we're familiar with: animals, plants, fungi, and some of you may have heard of protists before. The other cells, prokaryotic, the only types of cells that are ever going to be prokaryotic are bacteria-type structures. And actually, that group called prokaryotes was split into 2 domains that we call bacteria and archaea. And again, we'll talk more about that later. So for our purposes right now, it's just important that you understand the difference between prokaryotes and eukaryotes, which we're going to take a look at right now. Now, if we look at the similarities first, all cells, no matter what type we're looking at, has a plasma membrane. If you think about it logically, they need something to keep all of their insides in. That's the function of the plasma membrane. They have a semi-fluid substance called cytosol. You may have learned it in the past as being cytoplasm. Very similar structures there. All cells have some sort of DNA material, whether it's circular chromosomes or linear chromosomes, but they all have some sort of DNA material, and they all have ribosomes. And this is important. This comes back to the idea of going from DNA to RNA to protein, and ribosomes help with that process there. Now that's how we keep things similar between those two types of cells. Prokaryotes, on the other hand, have no nucleus. They have DNA, but it's not in a nucleus. It's just kind of floating around in an area that we call the nucleoid region. They have no organelles, and like we said, their cytoplasm is inside, bound by the plasma membrane. They're typically smaller than a eukaryotic cell, and they typically look something like this. On the inside of the cell, we don't really have much going on. Some DNA, that's the purple, and the dots would be our ribosomes, but that's about it. Cytoplasm, ribosomes, DNA, that's all really a prokaryotic cell has, and they're pretty small. Here is a picture of a pinhead with thousands of bacteria living on it. So they're a lot smaller than a eukaryotic cell, typically 1 to 5 micrometers. On the other hand, eukaryotic cells are larger. They're somewhere between 10 and 100 micrometers. And the reason that they're larger is because they have so much stuff inside of them. They have a nucleus, they have organelles like the mitochondria, the chloroplast, the Golgi bodies, the lysosomes, all of the organelles that you could possibly think of. And like prokaryotic cells, they have cytoplasm inside of the plasma membrane. And they're a lot larger, just like we said. Now for the rest of our time in this chapter, we are going to focus on eukaryotic cells, and we're going to learn all about the different structures that I just mentioned and organelles that I just mentioned, and you'll be able to know what they do, what they look like, and where they're located. Now the next topic is about surface area to volume ratio. I'm purposely going to skip this because we are going to be doing a lab where this explains it a lot better, and I don't want to give the answer away just yet. So these two slides, I'm going to skip them, and then we'll come back and talk about them later in lab. Now, one structure that we said that is common to both prokaryotes and eukaryotes is the plasma membrane. The plasma membrane is a lipid bilayer. It's made of phospholipids that you guys learned about in your biochemistry unit. And its main job, like we said, is to kind of keep everything inside the cell, keep it contained, and then it's also there being selectively permeable. It lets certain things come in, certain things come out, and it allows things like oxygen to come in, nutrients to come in, and waste to leave. Now, as I said, the rest of our chapter is going to be looking at the panoramic view of the eukaryotic cell. So we're going to go through and look at all of the different organelles that are inside of the eukaryotic cell, and we're going to do just a quick comparison of plant versus animal cells. So you have probably seen pictures like this. Back in freshman bio, you probably made the flip book. You did all kinds of little drawings with these. This is just going to be a quick review of all of those different structures that we've talked about in the past, and then we'll go a little bit more in depth with that. Looking at this picture, this is a typical picture of an animal cell, and this picture here is a typical plant cell. Take a minute, pause the video, and see what structures the plant cell has that the animal cell does not. Okay, so looking at what you found, and comparing those two pictures, you should have come up with 3 differences that the plant cell has that the animal cell does not. The first one, quite obviously right in the center, is usually some sort of big empty vacuole that we call the central vacuole. The chloroplast, whoops, I didn't want to do that. The chloroplast is another structure that the plant cell has that the animal cell does not. And our third is the cell wall. Right, we've talked about the cell wall being made of cellulose, that big carbohydrate. Those three structures are found in the plant cell, but not the animal cell. All of the other ones you should see are pretty common to both of them, but those three are the ones that are common in plant cells, but not animal cells. So in our next video, where we will pick up, is going through all of the different structures, the nucleus, the ribosomes, mitochondria, and so on, so that you are now familiar with all of the different structures of the eukaryotic cell.